Here's our second part about life in the soil. We're going to look closer into the microscope and talk about the different organisms in the soil. The first organism we're going to talk about is bacteria. Bacteria is the most abundant microorganism both in the soil and in the world. And part of the reason for that I'm going to explain is its growth rate. Bacteria can be both aerobic and anaerobic, so it can live in an oxygen-rich environment or an environment with no oxygen. If oxygen is available, oxygen is the terminal electron acceptor for respiration. If oxygen is not, sulfur is the terminal electron acceptor for respiration, for some other biomechanical process that makes energy for the bacteria without oxygen would be anaerobic, and that's when we get smelly, what my kids like to call pig mud, or anaerobic soil. So if you have really wet soil, when you smell it, that's what you're smelling. You're smelling that bacteria using sulfur as the terminal electron acceptor. They can be both heterotropic and autotropic. That means they can make their own food, or they can eat food, digest other things. They can be both saprophytes, saprophytic decomposers. Usually they're eating dead stuff. More than likely, sometimes there are diseases and things like that that are bacteria, but very few in turfgrass. And man has been able to use bacteria to cause some lesions on plants that have allowed man to do some genetic transformation of plants. So that would be crown gall, and that was one of the most early methods of genetic transformation. Not used as much anymore because we've come up with more sophisticated methods, but bacteria is very, very helpful and is used very much in the lab for transforming genes out, not only for the crown gall but also to produce genes to be later put in other plants or other organisms. So very interesting bacteria, very interesting classes. So microbiology is very important to understand this stuff. One of the reasons it's so important is it grows so fast. Here we start with 2 bacteria, 10 minutes later 10, 20 minutes later hundreds, thousands, hours later millions. So very, very fast production, reproduction. Bacteria multiply very, very fast. The cells just keep dividing. In fact, they divide so rapidly that the curve looks like this. We grow bacteria in any medium, and then at this point is where it runs out of food. So whatever food the bacteria has, it will start cannibalizing itself, and the population will go down. So hopefully this is a function of all populations. Hopefully humans haven't reached this point yet, but we have in history, and you'll see a dramatic fall off of populations and then a leveling off, or what we call survival of the fittest. So bacteria, because they reproduce so often, really are good examples of survival of the fittest and Darwin's theories of evolution and changing, and the best ones surviving and moving on. So a lot of that information we get about that for science comes from bacteria. The next group of microorganisms we're going to talk about are the fungi. And the fungi are important to turfgrass managers, especially because most of the diseases that affect turfgrass are from the fungi family. Not most fungi most fungi do not harm turfgrass. Most fungi are saprophytes. In fact, they're lignin decomposers, so they do a good job of decomposing the hardest things in the soil to decompose. They have structures called hyphae or mycelium. Pretty much for the course of this class, synonyms. I'm going to show you some pictures of those. All mushrooms are fungi. We can see here's some mushrooms growing on the side of a tree. They're starting to decompose the tree even while it's still alive. So that would be a parasitic mushroom, either coming up here from the soil and also getting on the tree. They release millions of spores. The undercap of a mushroom Produces lots and lots of spores. You can look at those under the microscope. That's fairly interesting. They have a large biomass. They're pretty big. They grow pretty rapidly, and actually can provide food for some humans that know what they're looking for. Don't just go eating any old mushroom. They grow a little better in an acid soil, so they're probably in a pine forest of North Carolina. Probably is a very good place to find mushrooms. Here's one of the turf troubles. This is what we call fairy ring. We're going to talk about this quite a bit in class next year, but no, that is a turf trouble. That is a fungi growing out in a ring, decomposing the stuff in the middle, mineralizing nitrogen to be greener here around the ring. Right here is just overabundance of hyphae that causes the soil to become anaerobic. So the plant doesn't die from this fungi. This fungi is a saprophyte, but the effect of excessive hyphae pulling moisture out of this area makes the soil so dry that the plant dies of lack of water. In some cases we just see the green area. It depends on moisture and how their disease is managed, or how the turf is managed also. The first discovered turfgrass disease was Rhizoctonia. We like to call it brown patch. This oily patch that we see here was originally called large patch because it's a large patch as opposed to small patch, which would be dollar spot. And one of the most characteristic things, and why I'd like to talk about Rhizoctonia, is the hyphae. So this is one of the most important things you need to be able to do with the microscope, is take some of this cottony mycelium that grows around the edges in the morning, put on a microscope slide, stick a cover slip on it, and if you have mycelium that is separated by these separations into T's, you can see that T there, you 100% have brown patch. And the chemicals for that will be different than some of the other diseases, particularly dollar spot. So you need to be able to diagnose this disease. Here's a close-up. This is what a slide looks like that we did in the lab a couple years back. Again, you can see that separated hyphae. So the hyphae looks almost like spider webs. In fact, sometimes it is spider webs. Throw it under a microscope, and if you have it separated like that into T's, you know that you have brown patch. Not all fungi are bad for turfgrass or plants. In fact, the majority of the fungi in the world are mycorrhizal, and mycorrhizal are symbiotic. So the host allows the bacteria, or in this case fungi, these symbiotic fungi, to live within its body, and in turn the plant gets an extended root system. And what happens is the hyphae from the mycorrhizal fungi go off into the soil, and they're very effective at taking up phosphorus. Phosphorus is a very immobile cation in the soil. It binds hard to the CEC. So this bacteria helps all the plants take phosphorus up, and we see plants grown without mycorrhizal do much worse than plants with this mycorrhizal symbiotic relationship. Unfortunately, almost all plants have this relationship, so we can only grow the mycorrhizal fungi in the lab. But we show that when we grow a sterile soil over and over again, even turfgrass and all plants do much worse without this symbiotic relationship of bacteria. And in lab next year, we'll show you some of the mycorrhizal fungi. 15% of the carbohydrates from the plant are given to the fungi, so that's quite a bit. That's a cost, but we would do the calculation that the phosphorus is worth that. They also can extend the root system for helping the plant take up water, and when they bloom and send spores out, they look like this. So your standard mushroom is usually coming from some sort of plant root, and it usually has a symbiotic relationship with that plant. Another fungi that is very important to turfgrass managers is the endophyte. The endophyte A, Coniphilium, you don't need to know that, but you definitely need to know in red here the term endophyte. An endophyte, like mycorrhizal, lives in the plant. Unlike mycorrhizal, it is viable in seed for a short period of time. So if you have endophyte infected seed, you want to plant that as quickly as possible. This symbiotic relationship with turfgrass is carried on to the next generation. This is unique in symbiotic relationships. What the endophyte does for the turfgrass plant is it provides an alkaloid, and that can be a problem to grass farmers. Grass farmers do not want to feed their cattle endophyte infected tall fescue because this alkaloid is so strong that it can cause hoof and mouth disease in the cattle. But turf managers that are managing fine sports turf or golf course turf or home lawns want the endophyte because this alkaloid is, as Dr. Funk, my mentor, said, an insurance policy for turf plants. It tastes bad. So an insect, given the choice of an endophyte infected grass or a non-endophyte infected grass, will choose the non-endophyte infected grass. So this is so much stress on the plant that a system, if you plant grass that's 10% endophyte infected, you come back 10 years later, it will all be endophyte infected grass because the pressures from the chewing, biting, sucking insects are so much that they will push the population to the alkaloid infected grass. So very helpful insurance policy and a very interesting ecosystem and symbiotic relationship that I was lucky enough to be able to study at Rutgers with Dr. Funk. The endophyte originated in Europe, and one of the big searches in breeding is to find endophyte infected other grasses. It is in ryegrass, it is mostly in tall fescue, and it is in some poa grasses, but not the poa grasses that we use. So Poa supina has it, and we're trying to genetically or some other way get this bacteria, get this fungi into other good turfgrass plants. Here's a picture of the endophyte. It lives in the cells, and we can see it with the microscope when we stain it with purple. It grows intercellularly. It's distributed, unevenly around the plant. So when the insect pierces that plant and gets a taste of that, it doesn't like it, so it goes on to something else. The most of the endophyte is in the leaf sheath. When the plant puts up the seed, the endophyte grows up and puts spores in the seed. Those spores do not last long, so endophyte infected seed has to be stored properly, and it has to be planted within 1 year. It forms a dense mycelium mat between the scutellum of the embryo and the starchy endosperm in the seed, and that can be transferred on to the next generation. So I give Dr. Reed Funk, this is Dr. Funk here at Rutgers, now retired professor emeritus, but a marvelous professor who spent pretty much all his time out in the field looking at the grass. And he figured this relationship out, and the work at Rutgers is still ongoing today to look at the endophyte and what it does to turfgrass. So the next microorganism we're going to talk about is the actinomycetes. The actinomycetes are one that probably most people haven't heard of before, but they're definitely my favorite. Neat system. A bacteria like fungi or a fungi like bacteria. Actually, they're bacteria that resemble fungi. They have an extensive hyphae system. You can see a picture up here of that hyphae system with many spores growing. They're like a little bit basic pH, so they're going to be found in a more basic soil. And when you smell what people say good soil that smells good and rich, sandy loam soil, what you're smelling is the actinomycetes. They like a dry, warm, slightly acidic soil. So they can go slightly acidic or slightly basic. So probably a little more basic than the fungi, but still a little basic, a little acidic is fine. They're saprophytes, so they eat the dead, dying stuff in the soil. They're very effective at doing that. So effective that one of them, namely called Streptomycin, and you've probably heard of that before, was effective at curing tuberculosis because of the nature of tuberculosis. In the next slide, I'm going to talk about that a little bit. When I was doing my master's degree at Rutgers, I had the pleasure of working at a place called Waxman Hall. It was named after these guys, and right across the street I also was able to work in a lab that they worked at. So these guys, Albert Schatz was a postdoc, A.B. Du Bois was a grad student, and Selman Waksman, you've probably heard of Selman Waksman because he won the Nobel Prize, was the professor. They were soil scientists. They solved the problem of tuberculosis. And if anybody knows any history of tuberculosis, if anybody's seen sanatoriums, this is where people were sent with this disease. If anybody's seen any old handkerchiefs from the 1920s, 1900s, there was always cherries or roses or some sort of decoration that was red on handkerchiefs. And the reason for that was because so many people were coughing up blood all the time. Tuberculosis was a disease that really devastated the population. And the problem with the disease is it would go into remission. And in remission, it forms a little bolus around itself, and that bolus is not easily attacked by medicine. So if you were to give somebody medicine that would normally kill a disease like tuberculosis, it wouldn't, because the disease would go into a kind of a hibernation state. Well, it turns out in the soil there's lots of organisms that go into this hibernation state too. Streptomycin was discovered in the cranberry bogs in South New Jersey, and it's an actinomycete, and it kills tuberculosis. So these guys worked that out. So it wasn't some great doctors, well, they were great doctors, I guess, but they weren't great medical doctors. They were great soil scientists. And if anybody wants to read The Forgotten Plague, I would encourage you to do that, because it's a very interesting story of how these guys, Merck Scientific, solved a problem that was a horrible, horrible disease, one of the #1 killers in the United States at the time. So on from my favorite microorganism to my least favorite. Really hate the algae, and I think in general algae does more damage to turf grass than fungi. There's 250,000 algae per tablespoon of soil. Algae is an aquatic organism that photosynthesizes on itself. So it's a very primitive organism, and pretty much probably was the first organism to emerge from the primordial sea. They're named for their color. There's green algae, brown algae, and red algae. Cyanobacteria is a blue-green algae, so it's not a bacteria. It has chlorophyll A, so it photosynthesizes itself. It's green. It makes its own food. First to evolve to come out of the sea. So it's the first organism to put oxygen in the air to make the Earth's atmosphere ready for other organisms that needed oxygen. Some of them can even fix nitrogen. The rate limiting factor for these bacteria is all phosphorus. So most turf fertilizers are low in phosphorus, partly because we don't want to get phosphorus in the pond. And if you get phosphorus in your turfgrass irrigation pond, you're making a mistake. And it's a big, huge mistake, and it's a mistake that kills lots and lots of putting greens. So you need to be very careful about your fertilizers. We need to control bacteria greatly in our ponds, rivers, and streams, or else they're going to look like this. And this is what we don't want. And if this is the irrigation pond for your golf course, I know you're going to have trouble, because that algae grows on the putting greens. When you water, you're putting it out there, and it's a big problem. So be careful with the algae. Control your ponds effectively, and be a good manager of the entire ecosystem. That's our microorganisms we're going to talk about. Next time we're going to talk about organic matter and something that goes on in these heavier soils. This is Bowie's Creek Golf Course in Keith Hills, Campbell University.